to form any kind of cells of the body. So if you have a person who has a disease or is missing a tissue or has an injury, you could regenerate tissue for them. And so that's what this whole idea of regenerative medicine is based on, is the idea that you could reproduce tissues that are identical genetically to the person getting them. So a big problem in transplant research, in bone marrow transplants, is that you need to have a genetic match or else your body will reject, and this would completely eliminate that. The other really big area of research is that you can take cells from a patient with a disorder of some kind. You can grow those cells in the lab and you can study them and you can try to understand what is the disease doing to these cells and how could we try to treat that. And so that's drug screening and other understanding the basis of disease progression. So just two short examples of that. So at Harvard Stem Cell Institute, some doctors are working with patients that have amyotrophic lateral sclerosis, or ALS. And in this case, they've taken skin cells, they've used the Yamanaka factors to make iPS cells, so exactly the protocol I've talked about, and then using a cocktail of growth factors that they know will trigger those iPS cells to grow into neurons and support cells. Now they can grow those in the lab. They carry the mutation that that patient has, and so they can now study these and try to figure out what is it that leads these to then degenerate and have problems in disease progression. Now, another idea behind iPS cells is gene therapy. So there has not been any gene therapy that I know of in humans. I may be wrong on